So the MASTERKEY-232 study was a study to assess the potential role of an oncolytic virus therapy called Talimogene laherparepvec, which is a modified type 1 herpes simplex virus vector containing a cassette that expresses the human granulocyte macrophage colony stimulating factor as a potential immunostimulatory gene therapy approach. In this trial, we looked to combine that therapy administered as an intratumoral injection alongside intravenous infusions of the anti-PD-1 monoclonal antibody Pembrolizumab. So this was a Phase Ib study, the aim of which was to define the safety and tolerability of the combination of T-VEC and Pembrolizumab in patients with relapsed metastatic head and neck cancer that was not suitable for surgical resection or curative reirradiation. Patients received injections of T-VEC intratumorally to a dose of 8 milliliters, maximum dose. And the initial dose was a relatively low dose of 1 million plaque-forming units per milliliter of the injection. Subsequent injections given 3 weeks later and then every 3 weeks consisted of 100 times that amount, so 100 million PFU per milliliter, again up to a maximum dose of 8 milliliters. The Pembrolizumab dose was the standard dose of Pembrolizumab given intravenously of 200 milligrams each time the patient received the dose, and that was every 3 weeks. The treatment was found to be safe and tolerable. We reported grade 3 toxicities in 3/4 of the patients. There was a single fatal dose-limiting toxicity in a patient who suffered an arterial hemorrhage. That patient had had prior irradiation and then re-irradiation of the local site and received injections of T-VEC very close to the great vessels in the neck and regrettably developed hemorrhage that ultimately proved to be fatal. There were no other grade 5 toxic events. In terms of the response to the therapy, we documented a response rate of 16.7%. This was not sufficiently high a signal to justify taking this strategy forward into subsequent studies. And so therefore the initial plan had been to take this into a randomized Phase 3 study. That study will not be activated as a result of these data.